I'm really pleased that Carol was able to come, and she's brought a little spring with us, so with her to us. So she comes from the University of North Carolina, Department of Marine Science, Earth?
Marine Science.
Just Marine Science. Where she's been since 1995, but before that she had lots of great adventures, starting off as an undergraduate in Wisconsin. Are you from Wisconsin?
Yes.
Okay. Lawrence University, and then she spent 3 years as a Fulbright at the Institute for Petroleum Organic Geochemistry in Germany, and then she went to MIT and the Woods Hole Oceanographic Institute program in 1987, and she got her PhD in marine chemistry, and her undergraduate degree was in chemistry. And she finished in 1993, and then she spent a year as a postdoc at the Max Planck Institute. But before that, I learned that she spent 3 months exploring parts of the Middle East that no one dares go near now, unless you'd have to. And then she spent a year at the Max Planck Institute before going to UNC. So her areas of interest and her specialties are in organic carbon cycling. And today, do you have your?
I have my title slide, which I can
All right.
there.
She's going to talk to us about.
I'm going to talk about carbon cycling.
Carbon cycling and microbial communities, and microbial activities and extracellular enzymes. So, without further ado, go ahead.
Is this on?
Yes. Oh, it is.
Okay.
You're good. Okay.
So I was first giving you a little bit of background. This is one of the field sites.
It was on. Okay.
This is one of the field sites that I have been fortunate enough to work with. This is actually the view from the front deck of the Marine Lab in New Ålesund up in Svalbard. So some of the data that I'm going to be presenting comes from here. Sort of fitting for the weather today. The weather in Svalbard isn't always this nice, but this was sort of late in the summer where the sun was just beginning to dip below the horizons, and we had clear weather, and this was like at 3 a.m. and it was just amazing colors. So that's why I have it on my desktop. Let me get up presentation. So I'm going to talk about the role of heterotrophic microbial communities in carbon cycling, and as a chemist I'm really interested in the relationship between the structure of organic matter and the ways that it is cycled by microbial communities. So trying to figure out this structure-activity relationship has been the major focus of most of my work, and this work has led me to spend a lot of time trying to figure out how to measure microbial extracellular enzyme activities, so that will be a major part of what I'm talking about today. So in general, what I'm going to discuss is a little bit of background on the marine carbon cycle. Sometimes when I talk to people, they say, you know, why would anybody care about microbial extracellular enzyme activities? It sounds like a fairly obscure topic. And one can view it that way, but it fits in a really important way into the major picture of carbon cycling in the ocean. And carbon cycling in the ocean has a huge effect on global climate and things like that, so it's important to put it into context. So we'll talk about the marine carbon cycle, some of the types of substrates that microbes consume. We'll talk about a little bit about a couple of characters that have been isolated in and sequenced in recent years that have given us some more information, and I'll talk about some of the challenges we've got in trying to measure the rates of microbial processes in the ocean. The second part of the talk is going to be mostly focused on data, some of the major results that have come out of my lab in the last couple of years, on contrasts in the ways that microbial communities process carbon contrasts both by environment and also by latitude. And I'll talk a little bit more in the final part, more speculation about the relationship between the composition of the microbial community and its function. Okay, Okay, so we know in broad outline how the carbon cycle actually functions. We know that CO2 here crosses the air-sea interface, and of course phytoplankton, this is my lovely artistic skills, you can see the phytoplankton photosynthesize and they turn CO2 into biomass. And then the biomass gets cycled through the food chain. That's supposed to be a zooplankton. I apologize, I'm a chemist, not an artist or a microbiologist or a biologist at all. And then gets cycled into the higher trophic levels. So all of these organisms process organic carbon, they excrete dissolved organic carbon, and we consider them part of the particulate organic carbon box. This bracket encompasses everything that effectively would be caught on a filter. So we filter ocean water. The stuff caught on the filter is POC. The stuff that passes through is DOC. And these little blops here and there at the DOC box and also on the particles and also in the sediments represent the microbial communities, the heterotrophic microbial communities. And their role is also to cycle organic carbon. They're responsible for a huge portion of carbon cycling that happens into the ocean, taking DOC, respiring it to CO2, and also repackaging it into sizes that can be grazed by small nanoflagellate grazers. These microbial communities also colonize the particles that sink to the bottom of the ocean, and they're responsible for respiring a lot of this organic carbon so that the particles that hit the sediments are actually only a small fraction of the huge portion that are made by phytoplankton and other organisms. Once you get down into the sediments, this carbon is gradually buried, except it's also worked on by the sedimentary microbial communities, which again produce dissolved organic carbon and CO2. And once you get kind of to the bottom of the slide, you get to some of my oldest work in Germany where we were looking at petroleum formation. That represents the tiny fraction of organic carbon that microbial communities are unable to cycle in modern environments that gets buried in geologic time. So if we wonder why should we keep funding ocean sciences if we're interested in what's going on in the world as a whole, one of the simple answers is that the ocean contains the vast majority of actively cycling carbon on Earth. Here, the size of the boxes in the left-hand figure are proportionate to the relative inventory, and you can see atmospheric CO2, which we talk about a lot, is actually about comparable in size to the seawater DOC box. Surface marine sediment organic carbon is also very active, and of course the marine dissolved inorganic carbon box is actually 8 times the size of the figure right there. So much of what's actually going on also in the atmospheric realm is driven by this huge box of dissolved inorganic carbon and what's happening in the other marine boxes. And if we take a look at organic carbon just in the ocean, it turns out that DOC actually makes up the vast majority of it. When I'm giving courses, a lot of times the undergraduates are amazed to find out, A, I'm not going to be talking a lot about major marine mammals because we're talking about carbon cycling, and B, on the total mass scale, you can see that the only organisms that make the picture are the phytoplankton, the zooplankton, and the bacteria. So these are living organisms. Most of the carbon in the ocean is actually detrital in one form or another, and the DOC box dwarfs all of the others. So the DOC is obviously a big chunk of total organic carbon. So you would think we know a lot about DOC. Many people have devoted their careers studying it. There have been numerous efforts to quantify it, to determine its dynamics, to determine its composition, and we've actually learned a fair amount. We know that dissolved organic carbon has a high concentration of combined carbohydrates. We know that lipids and amino acids and amino sugars make up another measurable fraction, but it's not a huge fraction. And when we compare the DOC of marine systems and of freshwater systems, we find that they're quite different in terms of structure. Freshwater DOC has a lot of aromatics. That is not the case for marine DOC. So we know that there are compositional differences between freshwater and marine DOC. We also know that the average radiocarbon age of marine DOC is surprisingly long. It's about 6,000 years, so that's about 3 times the total mixing time of the ocean. So if you think about it, the average DOC, whatever it might be, goes around through all of the oceans 3 times before it's somehow destroyed. That seems kind of odd. That's a long period of time. We know also that there's no average DOC per se. We can tell from various sorts of experiments that have been done in lab incubations as well as measurements at sea that have shown that we can operationally divide DOC into highly reactive fractions, semi-reactive or semi-labile fractions, and refractory fractions. But we don't know on a structural and chemical basis what actually distinguishes these fractions. So this is one of the main puzzles of the organic carbon chemistry of the ocean, at least from my perspective. So trying to figure out what's happening to this DOC box, and particularly what these players that can remove DOC and to some extent add DOC to the box will provide insight into a large chunk of what's going on in the global carbon cycle. So where do we look to examine microbial processing of organic carbon? We can look into the water column where there are these particles called marine snow, which are heavily colonized by bacteria. We can look also to the free-living bacteria in the water column. And then this picture right here simply shows a sediment core. This is from Svalbard, and the superficial sediments are oxidized. The lower sediments are strongly sulfate reducing. That's why they're so black from the iron sulfides. These are also areas of very active carbon cycle. If we want to think about who's there, as I'm sure you're aware, we don't have in culture representative organisms from much of the world's oceans, but a combination of new advances in culturing techniques plus what we've learned from genomics and metagenomics has helped us understand the capabilities of some of the organisms that we do have in culture. So this is simply one example. This is Pseudoralteromobis atlantica. Its genome has been fully sequenced. It's widely distributed in marine systems in the water column. It's also found on surfaces. It forms biofilms, and it's metabolically a very capable organism. It can hydrolyze a high variety of substrates, and it seems to be a very efficient processor of organic matter. So some of the insights that we've derived over the last decade or so, I would say, have definitely been informed by a lot of what we've learned in genomic investigations. This is another really interesting organism. This is Saprophagus degradans that was isolated from decaying seagrass, and this one turns out to hold the prize for degradation of polysaccharides. There's no other marine organism in culture that can handle as many substrates as this organism can, and getting it isolated, getting it cultured, and getting it sequenced took a while, but we are learning a lot about how marine organisms may operate from this particular character. In particular, it was believed that only terrestrial types of organisms built specific structures called cellulosomes, which are protein and enzyme containing structures with which an organism can take apart cellulose and lignin, for example. Well, it turns out that this particular organism also appears to make cellulosome types of structures, and that may help explain why it's particularly good at degrading a wide range of complex carbohydrates. Okay, but this brings us back to the more general question of what do we know about the way that microbes in the ocean select and access their substrates, and we're back to my cartoon illustrations. So we know that the organisms that we have in culture are not representative of everybody else that's out there. We do know some general principles about the way they operate, and one is that bacteria, gram-negative bacteria in particular, have an outer membrane through which they have these little inlet channels called porins. I've just put one on the membrane. They actually can make lots of them. The uptake channels, these porins, their dimensions set the limit for the types of substrates that can be transported into the cell. Typically, the limit is about 600 Daltons, which is equivalent to about a trisaccharide in terms of size. So any substrate that's bigger than about 600 Daltons has to be chopped up outside of the cell for transport into the cell. So here I have an organic macromolecule, and it's being hydrolyzed by exoacting enzymes right here that can clip off terminal units from large organic matter, and we have here an endoacting enzyme that can also clip off units mid-chain. So somebody in this community of heterotrophic microbes has to make the right types of enzymes that are going to chop up the organic molecule into things that are small enough to be taken up into the cell. So somehow the organism has to know what's there, the appropriate types of enzymes have to be made, and then the substrates have to be captured and transported back into the cell. So here we've got a bunch of problems. I already mentioned the microbiological and molecular biological problem that most of the organisms that are out there, we don't have in pure culture, so we don't know very much about their physiology. The problem that we've got from a chemist's point of view is we know equally little from a structural perspective about the substrates that they're using. So marine organic matter is hard to characterize structurally. It's got specific linkage positions, it's got specific components. Those are the types of information that you need to know in order to be able to determine what kind of enzyme is needed to take this thing apart. But if we take a look at the tools that we've got, in effect, we've got fuzzy magnifying glasses that tell us something about the components, but they don't tell us about the orientation of specific bonds. The other types of techniques that we've got essentially function as small bombs. We blow up the organic matter, we sweep up all of the pieces, and then we can make a really good catalog of what type of bricks there were. We've got 10 red bricks and 6 brown bricks and 3 yellow bricks. But what kind of pattern they made and how they were put together initially before we blew them up either with heat or with acid, we don't know. So this leaves us with a real dilemma as chemists in trying to figure out the structure of a substrate. What we do know is that most organic matter produced in the surface ocean is rapidly cycled. Not very much of it is preserved down here in the sediments. So the bacteria, the prokaryotes, the other organisms that are making extracellular enzymes have to have good ways of figuring out what kinds of enzymes they need. Otherwise, we would see more carbon preservation. So most of it is rapidly cycled, but why is DOC then so old? And what factors distinguish the organic carbon that is buried from the organic carbon that's cycled? And at this point, we then have to think about how do we measure enzyme activities in the ocean? How do we measure the activities of these tools that microbes use to chop up big substrates? The standard method that is used relies on small substrate proxies. Shown here, we've got a methylumbelliferyl fluorophore linked to glucose. And when this bond here is hydrolyzed, you end up with a big increase in fluorescence, so you can measure hydrolytic activity through an increase in fluorescence with time. The problem that you've got, though, is that this substrate is not actually a macromolecule. It's fairly low molecular weight. Martinez and Azam showed in 1993 that they can actually be hydrolyzed within the periplasm of bacteria. So you're measuring some combination of extracellular and periplasmic activity. The other problem that you've got is the more we study extracellular enzymes, the more we've learned about the fact that many of them have binding domains. So they basically have binding domains that hold a structure at the right orientation that the catalytic domain can actually hydrolyze the macromolecule. Small substrate proxies don't fit in the binding domains, so the enzymes don't work properly on substrate proxies. So there's a disconnect between the activities that we are measuring and the types of activities that are actually occurring in the ocean. So I've developed a different technique that I've used to take actual molecules, shown here as a strand of spaghetti, and add fluorescent tags to these molecules. So they're covalently attached. They remain attached to polysaccharides. I've done this with phytoplankton extracts and also with some phytoplankton DOM. You add these to seawater or you inject them into sediments, and then as they're hydrolyzed by extracellular enzymes, you end up with a bunch of fragments, some of which still have fluorophores attached to them. And you monitor the activity of enzymes by monitoring a change in molecular weight from a well-defined high molecular weight to a spectrum of lower molecular weight products. And if you know the molecular weight of the starting material, you know the size distribution of the final material, and you know the elapsed time, you can back calculate the hydrolysis rate. So the advantage here is that you can use true macromolecules. They'll accommodate the substrate binding domains as well as the catalytic domains. And you can measure the activities of enzymes that hydrolyze the macromolecules mid-chain as well as at the ends. So right after I developed this technique as a postdoc, I took a bunch of polysaccharides and fluorescently labeled them and went out on a cruise. And what you're looking at here are 6 different substrates that I had fluorescently labeled. This is polylin and laminarin. Those are both glucose-containing polysaccharides, but the glucose is put together in different linkages. This is xylin, which contains xylose, fucoidin, which is a fucose-containing polysaccharide, and arabinogalactan and chondroitin. And all of these polysaccharides are either marine-derived or the enzymes that hydrolyze these polysaccharides are known to occur in marine organisms. So what I had done was take these 6 different polysaccharides and I took superficial sediments and Skagerrak and mixed the polysaccharides in and simply monitored the hydrolysis rates with time. And you can see that all 6 polysaccharides were hydrolyzed with probably about a factor of 3 to 4 difference in hydrolysis rate. So the substrate structure does make a difference in how fast they're chopped up, but everything is chopped up pretty readily in these sediments. And just before we began doing the box coring here, I also took some water from a Niskin bottle cast that we had made. I had done all of my work to that point. The first 10 years of scientific research, I had worked in anoxic sediments. I'd never done anything in the water column, but I thought, I wonder what happens to these things when we throw them into the water. So from the bottom water overlying these sediments, I also measured hydrolysis rates. So that's what's shown here in red. So you've got the red axis for the seawater, the blue axis for the sediments. And I was really surprised by what I found, because what I found here, you can see the little red axis, the laminarin, the xylin, and the chondroitin were all hydrolyzed in the bottom water, whereas all of the substrates were hydrolyzed in the sediments. And you can see that the axes are offset by 2 orders of magnitude, so the difference in cell counts between seawater and sediments is typically 2 to 3 orders of magnitude in most places. So one of the obvious questions would be, well, is it that there are just more bacteria in surface sediments than there are in seawater? Well, there certainly are more bacteria in the sediments, but you can see no matter what I used as a scaling factor on the right-hand side, I would never get the patterns to match. There is hydrolysis of these 3 substrates here, and no matter how high I make the bars rise on this side, I'm not going to get the other 3 substrates to disappear. I was really puzzled by this, because if you think about what we learn about organic geochemistry and carbon cycling in the ocean, we typically picture fresh organic matter made by phytoplankton in the surface ocean, and as the particles sink through the water column, they're chewed on and worked over by various organisms. And by the time they get down to the sediments, the poor miserable buggers in the sediment have to deal with whatever is left over from the surface water. So you have low quality organic matter that just is not so desirable as a substrate. These data suggested that maybe the organisms in the water column actually didn't have the tools to take apart some of the substrates that could be sinking down, and maybe the organisms in the sediment didn't notice that their food had been chewed over because there was so much still available to them. But this was something that I wanted to look into. What I had found was a fundamental functional difference between the way the water column community acted and the sedimentary microbial community. So what could drive these differences? And at the time that I did this work, I wasn't able to find some of this information, but again, with the increased sequencing of quite a few, relatively speaking, marine microbes, it's possible to go wandering around in their genomes and start looking for sequences that correspond to what we think are these hydrolytic enzymes. So I took a look. This was not at the time I did the initial work, but this was about 2 years ago. I went wandering through the databases and took 3 marine examples, Gromella forsetti, Pseudomonas atlantica, and Rhodoparellium baltica, and I tried to find sequences that corresponded to the enzymes that would hydrolyze the substrates that I had been using. And I was able to find, here are all 6 substrates, and you can see that all 6 substrates are hydrolyzed by somebody, in case of fucoidin by everybody, but nobody can take everything across the board. So the interactive effects of having a whole community there with members that have different capabilities must be important, which means that the distribution of organisms in the ocean is really important. And when I did this is about the time that people began to recognize that there are biogeographical patterns also in the distribution of pelagic microbial communities. The old saying from Becking is that everything is everywhere and the environment selects, but we're beginning to get the picture, maybe not everything is quite everywhere in the ocean. We can see major patterns in biogeography. But in any case, with a couple of collaborators and numerous cruises of opportunity, we, over the course of a decade, were able to collect data from 32 different sites. This is up on Svalbard. These are specific data from cruises of opportunity, some work in the Gulf of Mexico prior to the Deepwater Horizon, and some work off the coast of the US, where we were able to incubate our polysaccharides in surface water and just see what happened to them. So what you're looking at now are the same 6 polysaccharides. This is the summed hydrolysis rate, where the size of each stacked bar contributes to the sum. And these are the stations that are organized by latitude. So this is degrees south in negative numbers. This is degrees north in positive numbers. And in the course of giving an overview talk, I finally put all of these data from a decade together, and I thought, no, by gum, it looks like there's a pattern here. We see that in the first place, laminarin is the only substrate that seems to be hydrolyzed pretty much everywhere in surface ocean waters. This makes a lot of sense. Laminarin is actually a storage product, an energy storage product for a bunch of different phytoplankton, including diatoms. So enzymes that can take apart laminarin are probably pretty widespread. Other than that, though, two different cruises in the Gulf of Mexico 6 years apart here showed the capability to hydrolyze all 6 polysaccharides in the water column. And this was the only marine location where we found this. All of the other locations, sometimes 5 of the substrates were hydrolyzed, sometimes 3, sometimes only 2, but there was a real distribution among them. And of course, looking at where all of the heights of these things fell, we thought there seems to also be a correlation between environmental temperature and some hydrolysis rates. So it looked like the enzyme activity has something to do with environmental temperature. Where it's warmer, you have high hydrolysis rates. But that doesn't really explain why we have these different patterns where the temperature is not so different. And when we did a little bit more sophisticated analysis, we discovered the temperature trend is driven predominantly by the fact that laminarin is hydrolyzed everywhere, and there's a close correlation between laminarin hydrolysis rate and temperature. So the fact that laminarinase is everywhere and it's closely linked with temperature pretty much drives the entire trend, plus the observation that at mid-latitudes there's a broader spectrum of enzyme activities than at high latitudes. And you can see that here, where polyolin is found mostly in mid-latitudes, and arabinogalactin and chondroitin are also hydrolyzed in different places. And if you take a look at the statistical distribution, the sum of the activities versus temperature relationship, 0.65, becomes better when you just focus on laminarin, and it actually is really lousy if you just focus on either chondroitin or on arabinogalactin. You can see that the hydrolysis rate at the same temperature can vary by a very large amount. So obviously temperature isn't the only thing driving all of this. So we find that some of the substrates are hydrolyzed everywhere, other substrates are hydrolyzed in only specific locations. This is particularly interesting because of the observation of semi-labile VOC. You might remember I had mentioned there's this fraction of DOC that seems to persist for a while and then it goes away. It's very possible that this semi-labile VOC is produced in a part of the ocean where the heterotrophic community lacks the capability to take it apart, and for it to be removed, it has to be moved to another part of the ocean that has a different microbial community with different capabilities. So the evidence for patterns of biogeography among microbial communities is particularly interesting. That's been coming out more and more in the last couple of years, particularly with more molecular biological investigations, there also seems to be evidence for reduced community diversity at high latitudes. So the microbial communities in polar regions appear to be somewhat less diverse than they are in more temperate regions. And you can't immediately jump from the diversity of the organisms to the diversity of the enzymes, but it suggests that there might be a tenuous link there. So with all of this, what about the microbial communities and sediments, which is what I initially started working on? Here we had to have not cruises of opportunity because they don't take the time to box core and collect your samples. We were just lucky to get surface water samples. So our sites are restricted to the areas where we had our own cruises and where we could do more detailed investigations. So what we found here is that all of the sedimentary microbial communities that we've tested to date are enzymatically very capable. Basically everything that we've put in, all 6 of these substrates plus a bunch of phytoplankton extracts that we've labeled have been hydrolyzed in the sediments that we've looked at. So why do we see these differences in patterns of enzyme activities between benthic and pelagic microbial communities? So one possibility is that there are differences in community composition between surface water and sedimentary communities. And when I initially did the work in the scatter rack, I thought that, after I got back from the cruise and analyzed all of my samples, I was going to go to the library and read all of the brilliant molecular biological studies where people had analyzed the microbial community in the water column, looked at what was going on in the sediments, and I could then make some sort of comparison. I was really surprised to find out that you couldn't do this, particularly at the time that I was looking for the data initially, because molecular ecologists tend, for the most part, to either work in the water column or work in sediments. So I could put together studies from different areas, a water column here, sediment there, sediment over here, water column over there, but I couldn't go to the same place in time and get data that had been produced in the same way. Slightly later, the first 2 studies were published. Hewson and colleagues in Hewson and Furman in 2003 and 2006. Hampton et al. Came out with a study last year. So there are literally only a handful of studies that have done this. Our study was actually the 3rd study. This was carried out in Smerenberg Fjord in Svalbard, one of my favorite places to work. This is Station J. So what we did is simply collect surface water, bottom water, superficial sediments, and then the sulfate-reducing zone, so like 10 centimeters into the sediments. That's deep in this particular figure. And then analyze the microbial community composition. I'm not a molecular ecologist. I'm not going to show you any trees. So this is simply a bar diagram showing you the major groupings of organisms that we found in the water column here: the surface water, bottom water, superficial sediment, and deeper sediment. And you can see simply by looking at the colors that the 2 water column communities, surface and bottom, resemble one another, and that they are quite different from the superficial and the deeper sediments. So the proteobacteria turned out to be really important in the water column. Both alpha and gamma proteobacteria, members of the Bacteroidetes, are here in the water column. They're also found in the sediments. There were some verrucomicrobia in the water column, and then the emergence of delta proteobacteria in the sediments. And if you go more deeply into the phylogeny of these organisms, you find that even where we had alpha proteobacteria in both cases, you had shifts in specific members of these communities. And the gamma proteobacteria, which are found in the water column and going into the sediments, the members of the gamma proteobacteria changed fairly substantially. So we found that there were major differences in the composition of these communities. We found that the alpha proteobacteria tended to disappear from the water column to the sediment. The composition of the Bacteroidetes and the gamma proteobacteria changed a lot. The delta proteobacteria first became apparent in sediments, and that the complexity of the clone libraries increased dramatically in the sediments compared to the water column. So in parallel to this, I had conducted more incubation experiments, in this case also including extracts of 3 types of plankton and alginic acid as a 7th type of polysaccharide. And I made incubations in the surface and bottom water, shown in the vertical and sort of slanted striped bars, as well as in the superficial and the deeper sediments to see what was hydrolyzed. And again, similar to what I had seen before, the polyolin was not hydrolyzed, the arabinogalactan was not hydrolyzed, and an extract of isochrysis was not hydrolyzed in either the surface or the bottom water. In the sediments, pretty much everything got hydrolyzed at fairly rapid rates. So it appears that there are functional consequences for carbon cycling in terms of what's used by these very different microbial communities when you compare the surface water and the sediments. So why would you have these differences between sedimentary and seawater communities? If you think about it again from an organic geochemical perspective, why pass up the opportunity to chow down on this fresh organic matter in the surface water and let it go all the way to the bottom where somebody else is going to have a chance at it? My thought on this, reading the literature and talking with people, is that there are probably fundamental differences in strategies of enzyme production and enzyme access between these communities, because of course there's an energetic cost in producing enzymes, there's an energetic cost in having larger genomes. There's a particularly nice paper, the Loro et al. 2009 paper, where they have compared different types of organisms. In the case of that paper, ones that are found in very organic carbon poor environments to organic carbon rich environments. They find striking differences not only in genome size, but also in the types of functions that these organisms can carry out in terms of their transporters, in terms of the types of proteins that are exported out of the cell, at least to the extent that the annotations are correct. So that suggests that the decision, if you will, for a microbe is in part how much baggage do you want to carry along in hopes of encountering a rich substrate, how much is it going to weigh you down in terms of your life and metabolic costs, versus how much of an opportunity are you going to miss if you're there with just one dull plastic knife and this large chunk of steak goes by you and you simply can't saw off a piece of it because you lack the right tools. So the whole genomic part of it, I think, is going to be a big part of the answer to what's going on. Some of it may also have to do with the diversity of microbial communities. Zinger and colleagues in 2011 did an analysis of a whole bunch of studies that had been published as part of the International Census of Marine Microbes. So they weren't taking samples from sediment and seawater at the same time, but they took tons of studies of near coastal, offshore, benthic and pelagic communities, all sorts of different types of marine environments, and found that there was greater diversity just looking at the community composition of the benthic as compared to the pelagic communities. And then, of course, there's the whole area where we haven't gone, the question of what is in the genome, to what extent does it get expressed. There may be very different strategies in terms of gene regulation for these different communities. So if we think sort of broad brush again, what factors impede organic carbon degradation by marine microbial communities? I suspect in sediments we're going to find that the communities are generally enzymatically capable. Their problems are going to be more issues of substrate access. How do you get at things that are stuck on the surfaces? How do you deal with problems of diffusion? How do you deal with problems that the enzyme that you release may end up being complexed or may end up being eaten by your next door neighbor who's decided not to work on the organic matter that fell down, but the stuff that you excreted? Those sorts of issues, I suspect, have a lot to do with what gets used and what doesn't. In the water column, I think that we're going to find that biogeographical differences in microbial communities have a much bigger role to play, and that either the enzymes are not produced because a cofactor is missing, because quorum sensing isn't satisfied, because a particular genetic pathway isn't turned on, or because a key organism simply is not present due to biogeographical differences in community organization. So what do we need to know moving forward? I would say one of the things that we need to know is how widespread these differences are in enzyme activities between sedimentary and water column communities. And also, do these differences in enzyme activities extend beyond carbohydrates and phytoplankton extracts to other classes of macromolecules? Here I have one final small study that I carried out a year and a half ago on Svalbard, where I took a small peptidase that's commercially available, leucine aminopeptidase, and then a couple of substrates for chymotrypsin and a couple of substrates for trypsin, to see what happens when you look at peptides of different composition in the water column and in surficial sediments. And in this particular study, I worked in 2 different fjords, in surface water and in bottom water. So here's the surface water, 2 different fjords. Here's the bottom water, 2 different fjords, and then the surficial sediments from both fjords. And I found that in the surficial sediments, all 5 of these substrates pretty much went like a shot, but the same substrate wasn't used in the bottom water or in the surface water of either of the fjords. I know from other studies that people have been carrying out that in temperate environments, all of these sorts of substrates have been hydrolyzed. So this suggests that there is a benthic pelagic contrast at high latitude that may not exist at lower latitudes. So we may have biogeographical patterns and latitudinal gradients in peptidase activities as well. So from a microbial perspective, structure is a super important parameter in determining organic matter quality and the extent to which it can be accessed. But I think as organic geochemists, we have to come away from the idea that we can make a good definition using one set of rules or one set of parameters, because the capabilities of the microbial community play a huge role in determining whether this organic matter is going to be metabolized or not. The capabilities, I think, are directly linked to the composition of the community. So where should we go from here? I would say we need a biogeography of the marine carbon cycle where we get a better handle on the biogeography of communities and of their capabilities, and where we make further progress in efforts to structurally characterize organic matter in ways that go beyond cataloging the building blocks and give us more information about bond structure and things that we can use in terms of analysis of enzyme susceptibility. So with that, I want to acknowledge many people who have contributed to the success of this work, primarily Sharif Gobriel, my lab manager, and Kai Siervogel, who's a research associate at UNC, Andrew Steen, who finished up a PhD with me and is now at the University of Tennessee in Knoxville, Wade Jeffrey, who went on a lot of cruises of opportunity, and many colleagues at Max Planck Institute in Bremen, where Josie didn't mention it, but I've been at Carolina since 1995, and I've figured out I've spent 4 or 5 of those years working in Bremen on leave, on sabbatical, on don't tell my department chair that's where I am again. So it's been scientifically and personally a great relationship for me. We've also had a lot of help from scientific parties, captains and crews, and of course the funding that has helped make all of this possible. So thank you very much. Questions?
Yes, can you talk, your experiments are fairly short term, and you haven't really talked about what sort of the recent history of that water and sediment might do to the rates that you measure.
That's where, particularly in polar regions, being able to do seasonal studies would be totally critical. The incubations with the labeled polysaccharides actually extend long term, since you have to see a change in the molecular weight of the added substrate. They can go for 3 or 4 weeks, and in the case of polulin, I was so surprised that it wasn't hydrolyzed. One of the semesters that I was on leave in Bremen, I took the samples back and I let them incubate for 6 months, and I still couldn't find hydrolysis. But I agree with you, things may change, particularly in polar regions, on a seasonal basis, but I haven't had the opportunity to find out.
So in the shallower regions, for example, like you were showing the pictures of in Sweden, wouldn't a lot of the water column particles actually be resuspended particles from the bottom? And given that, why would there be such a difference in the expression of enzyme activity and microbial activity associated with that kind of material?
For the work that I did in the Skagerrak, that was actually it was one of the deeper parts. That was 220, 230 meters, I think. as I say, that was the first time I tried this, and we were about 10 meters off the bottom because I wanted to not have the CTD and the just get bottles, bang the bottom and stir things up. There's a lot of information about organic carbon in the Skagerrak that it may be laterally transported from other areas of the North Sea, but I think we were high enough in the water column that we weren't getting it. For the work that I've done on Svalbard, the main station that we've worked at is also in a fairly deep fjord, so the depth at that station is about 218 meters, and again, the bottom water is about 210 meters. There's no terrestrial vegetation to speak of on Svalbard. The biggest plant is about that big, and about 95% of the landscape is fully covered by glaciers. So you do get, in some of the fjords, significant input of glacial flour, but that doesn't seem to add much other than turbidity to the water column, and it apparently impacts the primary productivity a lot, but not what's happening in terms of the sediments. So I don't think there's much sedimentary suspension in the areas that we've worked in.
I'd like to ask one more question, please. That when you showed the latitudinal patterns,
Yeah.
there was a asymmetry,
Yes.
north-south asymmetry, and I'm wondering if you could comment on what you think contributes to that. I mean,
Oh, I think what contributes to that in the first order is where we had cruises of opportunity. So we have totally not at all evenly spread out patterns. So a lot of the stations were right down here. Jeffrey was aboard one of the resupply ships going down to McMurdo. So they had a whole bunch of stations down here. You can see we don't have all that great latitudinal coverage. It was interesting that this point right here fell in with the 2 points on Svalbard. So one of the things that amazes me about that study is that we saw any pattern at all, given how varied oceanically these stations are. So yeah, if I get funded, we'll have much better geographical coverage.
sure,
Check with me in a couple of years.
A lot of your stations center on what would be called active margin regions and in the middle of the oceans, and very few, except for the northeast U.S., are associated with.
These were funded, those were funded, these were funded by the Max Planck Institute. These were 2 cruises of opportunity, and this was 2 cruises of opportunity. So.
I wanted to address that same point. Today in class, we discussed what besides temperature, because you said there were probably other things. And one of the things we noticed is that there's also a correlation between land and, higher land mass and the higher rates.
And actually, one of my colleagues in physical oceanography was asking for help on a proposal. I just have to get down to this figure that he made for me. This one. So he pointed out that there's a correlation between where we have the subtropical gyres. He just did the Atlantic for me. We have sort of parallel patterns of subtropical gyres in the Pacific. So here are the subtropical gyres, and here's the polar gyre and the Antarctic circumpolar current. And he divided up the patterns by all of that, suggesting, it may have to do again with biogeography because of the patterns of current circulation. So I thought that was really cool. So this figure was made by John Bain.
Well, one of the things is if there is a land influence, there's been a lot of controversy about how much terrestrial organic matter gets out in the ocean. You've chosen primarily marine sugars. If you could do the same thing with terrestrial sugars, make fluorescent labels of terrestrial sugars, and do that same latitudinal thing, you might be able to shed some further light on that question of terrestrial versus marine organic matter.
That's interesting because we've worked a little bit in river mouths in the Chesapeake and in the Delaware, and we didn't put stations that were non-marine by salinity in here. And in most cases, we have a greater spectrum of activity in the riverine stations than we do in the marine ones. And I should also mention that xylem is also found in terrestrial plants.
But you could choose more representative sugars.
Yeah, we could pick other sorts of polysaccharides. So those are only marine stations where we had full marine salinity.
Yeah, I have a question that relates a little bit to the first 2 questions. What is your vertical resolution of your measurements? So you mentioned surface water, water column, above the sediment, in the sediment, and wondering if you see there's meters or centimeters, if you see huge gradients and then diffusion effects. And why I'm asking that is, if I want to understand DOC in the surface waters, which may become airborne again, do I have to consider the remaining, the sediments or the entire water column to have a good description of the organic matter in the ocean surface water.
So the water column, in each case, it was a Niskin bottle where we had to deploy it deep enough that it was below the surface, so say 2 meters. The bottom water, in each case, we didn't want to be resuspending sediments, so it was typically 5 to 8 meters off the bottom. In the sediments, it depended on the depth of oxygen penetration, so we used the oxidized surface sediments, which was about the first 1.5 centimeters, and the deeper sediments from other work that we've done with sulfate reduction rates. We knew we were in the sulfate reduction zone maximum, so it was like 3 to 9. With the sediments, you can increase the resolution. These measurements were done by adding the substrate to homogenized sediments, but I've also done in parallel to what people do to measure sulfate reduction rates, where you have a core that's got little injection ports and you inject the radiolabeled sulfide. You can take the same types of cores and inject the fluorescently labeled polysaccharide. You then let it incubate for a while, and then I section the core and get the pore water out. And because of both the volume that I inject and diffusion, I typically find you get like 2 centimeter sections. So if I inject it in the middle of a 2 centimeter section in a typical muddy sediment, I can capture all of that substrate getting out. It's not millimeter level resolution in sediments.
But that's but the chemistry of the organic matter, is that now affected by such a gradient from the surface waters to the sediments? Or, no, that's maybe a naive question, I'm not a marine scientist, but I'm interested about the organic carbon in the sea surface water.
Well, I mean, other people's work in studying DOC, there's actually some of Cindy's work and some of her colleagues, there is a distinct microlayer at the interface between the atmosphere and the water column, and that has compositionally different organic matter than the stuff below. But for what I'm looking at, I'm kind of looking at the bulk things.
On this data again, I just want to be sure, did you normalize for population density of the microbial community?
No, we don't have cell counts for that, but again, knowing broadly how cell counts vary again from the census of marine microbes and things like that, you can't get an explanation out by picking any scaling factor for microbial communities.
I have another question. You showed the population, how much bacteria you have in the column, and then compares the
Compared to the sediments?
sediment. What method did you use to get that?
So those were clone libraries that we had. So how I did the measurements, we were out there on this very small research ship, so I was up in the bow where we weren't throwing mud around, hand pumping things through filters that we then did the molecular biological PCR amplification and all of that sort of stuff to get them out. And then I went back to the store and started my incubation stuff. So.
Yeah, I'm interested in the ability for exoenzymatic attack of particulate versus dissolved organic matter. And if you're a microbe, and whether you're in marine snow or in sediments in these relatively oligotrophic areas, you're in dilute solution. And if you're going to throw out enzymes, you'd like your substrate to be close to the boundary layer of the cell,
Right.
which might be one reason why sedimentary organisms really like to use enzymes. And there's a big pool of material in the particulate phase. Your probes, I don't completely understand the experiments, are relatively soluble.
They have to be soluble, otherwise they get liquid.
Polysaccharides.
Yeah.
Is there what do we know about the relative activity if they were absorbed versus in pore waters? Some of yours are going to be more particle reactive than others. You have other experiments. Do you have a sense for whether you get a faster activity if something is on the surface as opposed to?
We did some work on this. It was a study that was published in, I think, 2007, 2008. So the substrates that I used to get the labeling to work, it has to be soluble to have everything characterized. What we did, though, was take some of these labeled soluble substrates, and we then chemically tethered them to these little beads so that they couldn't diffuse all over the place. And we did comparative experiments of hydrolysis of the labeled polysaccharides that were tethered to beads with we were using purified enzymes in some cases and natural microbial communities in others. So we had the enzymes that were free to attack the beads. We also took the enzymes, absorbed them to mineral particles, and then mixed absorbed enzymes and the tethered substrates on wave tables to take a look exactly at some of those factors. So our observation was that the size spectrum of what we got hydrolyzed changed whether, depending on whether the enzyme was stuck to a particle or free in solution. The fastest hydrolysis happened when the enzyme was free in solution and could get at the tethered substrate or get at the free substrate most easily, then getting at the tethered substrate. But the principal difference that we found was that the size spectrum really changed and the lifetime of the enzyme changed when we absorbed it. So it lived longer, if you will, when it was stuck on the particle, and it produced a different size spectrum of products. It's a really interesting question getting at how does particulate organic matter get hydrolyzed away. That's one of the reasons that I showed the saprophycans picture, because what we found, not me, but the folks who've worked on them, what the folks who've worked on these organisms have found is that they preferentially stick on particles and apparently erect basically structures that organize the enzymes in ways that, as I envision it, they can scrape off what's there on the surface and retain the hydrolysate within a fairly small volume. I mean, it's a strategy that's also known from terrestrial systems.
One more.
I just wanted to change the subject completely. Your work and all the questions so far have concentrated on the production side. Now, have you thought anything about the loss side? I was thinking here as I saw your pictures that in the water column you might have more generalized predators, and the only thing that would survive is the thing that grows fastest, and this might be something that's growing on the most abundant substrate. Perhaps in the sediments there are different sorts of controls. For example, you might have specific viruses or something. And it could be that all these patterns are due to the loss side rather than the production side. How much have you thought about that, or other people thought about that?
That is a really interesting question. There was a very cool paper, I can't remember exactly where it was published. The commentary was by Dave Kirschman, and the original paper was out of Steve Giovanotti's group, and it had to do with SAR11 bacteria. So they're members of the alpha proteobacteria. Their genomes have been sequenced. They don't make extracellular enzymes, but one of the big issues is we find members of the SAR11 all over the place. And initially the suggestion was maybe they're viral resistant and that's why they're so abundant. This really cool paper showed that actually they're not viral resistant. There are other strategies that probably make them successful. I think you're right, particularly in organic rich environments where you have more viral particles because you have higher concentrations of microbial cells. We should think about stuff like who's, the protozoan who were grazing on these bacteria, what effect did they have also on the patterns that we're measuring? We haven't done any experiments in that direction, but I think it's a great question and a really important issue to consider.
Cindy has one more.
There's one more question which I'm going to ask on behalf of our very shy students in scientific communication. One of the questions that came up, I think you want to ask, it's very interesting, pH. You never talked about pH in your papers, and of course enzymes are very sensitive to pH, and like surface water and deep water would have very different pHs. What do you think about that influence?
I suspect in this particular case they probably don't, it probably doesn't have that much of an influence because you're correct, the absolute rate will vary according to pH, but the pH gradient is probably not huge in the bottom of Smearenberg Fjord compared to the surface of Smearenberg
But on your
Fjord.
latitude, no.
These
Oh,
are all
you did
surface
active
waters.
Surface. Okay,
Yeah, these are all
surface,
surface
yeah.
waters. So,
But sediments, your sediments will differ greatly in pH.
yeah, that's also possible, but I think what we're focusing more is on the difference in patterns rather than, there was this rate 10 and that one's 14
Do
and
you
how do they
measure pH
explain.
when you do these experiments?
We haven't, no.
Start.
We have started that, since we've started doing river work, we've started doing it because of course there you can't figure gigantic carbonate buffer and small pH changes. And we actually have a manuscript in review on some lakes that we had an opportunity to sample, and the pH variations there were insane. They dropped by, one of the lakes changed by 1.5 pH units from the spring to the late fall. So that, a couple of years ago, totally woke us up to the fact that we need to be more careful about this when we work with pressurized systems.
Okay, well, we can continue discussion at lunch. Thank you.